Okay, so welcome. We are moving on to our next topic here, our next biomolecule, really, which is proteins. We just finished up with nucleic acids. Now we want to learn more about proteins, and I have some images of proteins down here. They're highly complex, coiled, as you can see. They're very big. Here's a picture of some amino acids strung together, kind of a more local level, a closer look in at this protein. But we'll talk about this more in depth. So, this is the most structurally and functionally diverse group. Remember, RNA came first, then DNA. If you don't remember from our evolution video, DNA was more adept at storing information, so it was next to evolve. And proteins were more adept at doing things, doing a lot of different things. They're involved in almost everything in your body. Enzymes. Enzymes speed up chemical reactions, things such as pepsin, which this is an image of over here. DNA polymerase, which is the enzyme that kind of puts DNA nucleotides, which we talked about last video, kind of hooks them together and helps a growing strand of DNA grow. Structure, keratin, which is in your skin and fingernails. Collagen is in your hair. This is a very, this is a structurally important protein for our body, right? This is these long strands that we can combine together. Carriers of transport, things like hemoglobin, which you've probably heard of before, and aquaporin. Aquaporin transports water into and out of your cells. Hemoglobin obviously transports oxygen throughout your whole body as your blood supply. Here's a picture of hemoglobin down here. Hemoglobin, right? Collagen. Here's pepsin. Label these as we go through. Cell communication. Things like insulin. A lot of these things that tell your cells to do certain things. This is insulin over here. It's labeled down here. Human insulin and pig insulin. So signaling molecules, things that can tell cells to do things. Also, receptors, the receiving of signals. They operate in defense. Antibodies. You've heard of antibodies before. I'm sure this would help in cell recognition. Cells to recognize other cells. These are some antibodies down here. They have this very unique Y shape. Movement. Actin and myosin, right? These are more structure proteins, kind of like collagen. Well, they are more complex. These operate in muscle movement, right? Very, very important to us. This is actin and myosin chain over here. Here's a myosin and actin. Okay. So, as you can see, they do a lot of different things. They are very important in our body for, also it's not mentioned here, but I think just a general way to think of this is growth and repair. Proteins are hugely important for that. Okay, so let's take a look at some structure and what these proteins are made of. Well, a monomer, remember this is that word for a single unit of these, are called amino acids, which we'll study in depth, I believe on the next slide here. There are 20 different unique amino acids. Compare that with nucleic acids, which had, for DNA, it had 4 different bases. If you include RNA, there's 5 total with uracil. Proteins have 20 different amino acids, right? It's kind of similar to our alphabet. So you can come up with you can say a lot of different things with these amino acids. And the polymer for proteins, it's called a polypeptide. Obviously, you see this word poly meaning many, right? And peptide is kind of this refers to these amino acids that are going to chain together. Protein can be one or more polypeptide chains folded and bonded together. Again, these are large, complex molecules. They have a complex 3D shape. That's what makes them so important, is their ability to fold. So let's look at the structure of a single amino acid. Again, if you remember from an earlier video when we talked about functional groups, I said that R just kind of meant stuff. In this case, for amino acids, these are what we are going to call side chains. Which we'll look at on the next slide here. But the basic structure of every amino acid, again, is a central carbon. Number 1, I'll circle that. The central carbon right here. Right? The amino group, right? We talked about functional groups. The amino group is in blue here. Amino group. A carboxyl group, or a carboxylic acid, over here on this side. Let's look at it. Carboxyl group. Again, this is our central carbon here. Carbon. right? And then we have an R group or side chain, and this is where things vary. This is how we get our different amino acids. Every single amino acid has these three things that I'm underlining here: a central carbon, amino group, and a carboxyl group. Every single amino acid also has a side chain, but this side chain is unique. This is where they vary right here, is in these side chains. So this is our variable group. For each amino acid, the side chain is different, and it gives each amino acid unique chemical properties. Well, things such as, you've heard this before, hydrophobic, hydrophilic. We'll look at some of these on the next slide. Hydrophilic property, these types of things, make them interact and fold differently. Okay, so what I'd like you to do is go ahead and pause the video. Let's look at this. This is tryptophan right here. This is my favorite amino acid. I don't know why, I just like it. I want you to go ahead and put a star next to the central carbon, and then circle the amino group, and circle the carboxyl group, and find the unique side chain R. Okay. Good job. All right, let's see how you did. Did you, your central carbon for this molecule, again, remember there's carbons all over the place, is right here. It's right here. This is your central carbon. Your amino group is this down here. This is your amino group. Over here, you have a carboxyl group. Carboxyl group, or a carboxylic acid. And then all the rest of this over here, right? This whole big double ring structure, this is your R group. Side chain. This is what makes this amino acid unique. This is what makes this amino acid tryptophan, right? A very specific amino acid. But it still has all the central carbon, carboxyl group, amino group. The only thing that's going to be different from other amino acids is this R group. Okay, so here's an amino acid chart. I think this has all. You can count them if you want. 5, 6, 7, 8, 9, 11, 12, 13, 14, 15, 16, 17, yeah, that's 20. There's all of them. No, there are other amino acids that our body can make, but they have to be specially synthesized. These are our general amino acids here, and there are 20 of them. So they have different properties. Some amino acids have electrically charged side chains. Right? These are side chains that have an electrical charge to them, negative or positive. Right? Typically, it involves again these highly electronegative atoms, right? Either having an extra hydrogen or having an extra oxygen over here. Amino acids with polar, uncharged side chains, right? So these are side chains that don't have electric charge, but they are polar. So you can look on here and tell that these have polar molecules in them, things like nitrogen, oxygen, all of these. There's some special cases. These are special bonding that's involved in glycine, proline, and cysteine. Glycine is very small. It's the smallest amino acid right there. All right, amino acids with hydrophobic side chains. This means these are not polar side chains. And you'll notice there's a lot of carbon in these. Not a lot of polar molecules like this. I mean, there's some, but not a lot of them. So these are hydrophobic side chains. So this is going to affect how each of these proteins, when you have a long string of them, how they're going to fold and what they're going to look like when they fold. Of course, hydrophobic things are going to want to stay away, right, from kind of the water contents of the cell and face each other. Polar side chains are going to want to face towards water, right? And these things are going to bond to each other in very special ways. As we're going to see. So, final thing I'm going to talk about for this video is I want to show you some dehydration synthesis again. Peptide bonds. I want you to recall right now. Go ahead and pause the video and try and recall what the bonding between the phosphate group and hydroxyl group in nucleic acids is called. What is that called? Okay, you should have thought of phosphodiester bond. I'll write pretty small up here. Phosphodiester bond. This is just seeing if you remember this. Diester bond, and of course that is in nucleic acids. So peptide bonds are what we call these bonds that are in proteins, between amino acids and proteins. Okay, so this is a covalent bond between the amino group of one amino acid and the carboxyl group of another. So you can see over here, you have your carboxyl group, right, and you have your amino group. And when you remove water or dehydrate it, again, this is the water OH, these two H's in the O, this gets removed, and you end up with this polypeptide chain. Again, this R group could be very different. You know what that is. For the sake of simplicity, they haven't put our group on there or shown you what it is. But they've shown you kind of what happens when you bond two amino acids together. You end up with with a growing polypeptide chain. This is called a peptide bond. There's a bond between a carbon and a nitrogen, and this is the process and action of dehydration synthesis. So I'm going to go ahead and stop this video right here, and we'll continue talking about proteins in the next video.